Hello everyone, welcome to my channel Microbial Community, and today my topic of discussion is antibody drug conjugate. So in the introductory part, uh, what is antibody? An antibody is also known as immunoglobulin, is a large Y-shaped protein used by a immune system to identify and neutralize the foreign objects such as pathogenic bacteria or viruses or simply pathogenic microorganisms. The role of antibody is uh, to terminate the activity of the pathogenic microorganisms or pathogenic substances. And this antibody is we also call this as a immunoglobulins, and these are the Y-shaped structure. And then comes to the what is antibody drug conjugate. As the name suggests, that antibody drug conjugate, that means antibody needs to get attached with a drug or conjugate with a drug. So this antibody plus drug is called as a antibody drug conjugate. But here the point to be noted is that antibody directly cannot bind with the any conjugate or any kind of cytotoxins or any kinds of drugs, uh, but antibody can link with a drug with the help of a linker. So linker is a very important part in the antibody drug conjugate, and these antibody drug conjugates are basically useful in the production or useful in the termination of the cancer disease, or it usually helps in the treatment of the cancer. So in what is cancer cells? Cancer cells are the cells that grow like uncontrolled growth, cells that have uncontrolled growth, those are called the cancer cells. And one of the main reason of the cancer cells is the defect in the tumor suppressor genes, and these tumor suppressor genes has a proper tumor specific antigens and ADC, or the antibody drug conjugate, that basically attacks onto that onto the tumor specific antigens. And here the antibody that is used, that is monoclonal antibody. So moving on to our next slide, structure of antibody drug conjugate. So as I already said that in the structure, monoclonal antibody is linked with a conjugate or a drug or cytotoxins with the help of a linker. Here linker is one of the important substances or important part in the antibody drug conjugate structure, because if the linker is not properly worked, this drug can be released into the normal cells or any time in the blood or any time in the body when it is circulating. Uh, so linker is a very important thing. Earlier days, when the antibody drug conjugate is concept is just going to is just discovered, and they are preparing the researchers are preparing the proper antibody drug conjugate against the cancers. At that time, the antibody drug conjugate complex cannot be able to internalize. uh, Nowadays the scientists find a reason, or scientists go for a internalization of the monoclonal antibodies plus drug plus uh, linker. Whole ADC complex can be able to easily internalize. And see, the main reason is that first we are using, first the scientists or researchers are using the chimeric and monoclonal antibodies, or that monoclonal antibodies that usually makes from a like different species or mouse. In case of chimeric, the constant regions are from the mouse, but the like variable regions are from the humans. So that are called the chimeric antibodies. So that's how they are making antibodies. But nowadays they are making proper human monoclonal antibody, no mixing, nothing, only human and monoclonal antibodies. with they are attaching that human monoclonal antibody with the linker and the proper cytotoxins or the proper drugs, and then they are intravenously included it inside our body, and that things helps in the internalization, proper internalization, and for the proper activity of the drugs, and for the proper activity of this complex needs to get internalized. In the next slide, these are the main points, what are the tumor antigens, antibody, linker, and payload. The tumor antigens are the it is abundant in the tumor cells, minimal in the normal cell. These get internalized upon binding to the ADC. Antibody. So antibody. Here the antibody is monoclonal antibody, as I already said, because of their high affinity towards the antigens. And one of the main part is the linker. See, for one of the first drug, or one of the first ADC, that is FDA, that was FDA approved. This drug was later disapproved. The name of the drug is uh, gemtuzumab ozogamycin. That is already written here. You can see that, and this drug is later disapproved by the FDA because of they found that this drug has the defect or the problem in the linker. They are releasing the drugs inside our body, not specifically. They are releasing the bonding of the linker and the drug is very loose, not specific, and this drug is releasing in any time, and they are causing the disease in the normal cells also. In the normal cells, they are affecting the normal cells. That's why this leads to serious kind of hepatotoxicity. This linker is very important thing. And payload, here payload means nothing but cytotoxins or cytotoxic drugs, or any drugs or cytotoxins we call it as a payload. So as you can see that it is already written that drug cytotoxic to targeted tumor cells, not hydrophobic, must be potent since limited number of the molecules can be attached to the antibody. So in the next slide, this is the whole structure of antibody drug conjugate. In this structure you can find, this is the this there is the labeling that you can see that this is the monoclonal antibody, and this is potent cytotoxic payload, and this is stable linker. That linker helps in the attachment of the cytotoxic payload or cytotoxins to the monoclonal antibody. And basically those are not uh, specific that these cytotoxins should bind to the CH2 or should bind to the CH3. They can bind in the CH2 or in the CH3 domains, but not in the CH1 or variable region domains. So that is the one of the main point to be noted. And in the next slide, this is the evolution of the chemotherapeutic drugs. So in 1958, methoxytrexate, you can see the name, an anti-metabolite or antifolate, cures the first solid tumor using the chemotherapy. And in the and also dexorubicin, and in the year of 1960s to 1970, zinc alkaloids, and camptothecin. Now, there are several types of, according to the ages, like in 1950s to 1990s. There are 1990s or 2000. There are several kinds of drugs that were helps to treat or that was helps to treat the cancer disease. But in the year of 2000, first ADC or first antibody drug conjugate that is approved by the FDA, that is gemtuzumab ozogamycin, approved by the US FDA to treat acute myeloid leukemia. So that is the one of the main point to be noted, but later it is disapproved, but it is the first FDA approved ADC, and now it is also disapproved. And in the year of 2013, 2013 third ADC is approved, like you can see the name, trastuzumab M-tansine treats the HER2 plus breast cancer. It is the first ADC to target a solid tumor. First ADC to target a solid tumor. That is the one of the main point to be noted. So these are the evolution of the chemotherapeutic drugs you can find here in the next slide. This is the full mechanism and what are the challenges in this ADC making of the ADC or the function of the ADC. So in the full mechanisms you can see first is the circulation. ADC solution is prepared and released into the bloodstream. As I earlier said that this whole condition is the intravenous conditions, or it's like an injection. We need to put it inside the body for the proper function. So a proper solution is made, like proper antibody drug conjugate is made, and this needs to be needs to get inside the body. And what are the challenges in this? A stable linker is required to limit non-target toxicity. As I already said, the linker is one of the important thing in this whole process. So if the linker is not properly attached, it can release in any time. It can release any time, and that leads to a severe diseases to the cancer patients. So in the second step, and this is the binding step. A monoclonal antibody component of ADC binds to a tumor-specific antigen. So, first when it is pushed inside the body or intravenously it is pushed, then first this solution or the total ADC complex is binds to the tumor-specific antigens that are present on the tumor cells, and that will bind to that specific antigens. This is the proper binding needs to be there. So for that, monoclonal antibody must retain a high immunoaffinity. That is very important thing. That it needs to have a high immunoaffinity towards the antigen. So in this third step, internalization process occurs, and in this the ADC antigen complex undergoes receptor mediated endocytosis. In this step, this whole ADC and that binds to the tumor specific antigen that needs to go for the internalization process, and they tries to form a endosome. They start upon circulation, they tries to form a endosome. So here what is the challenge in this step? Inefficient internalization and limited target antigens prevent the cytotoxin from reaching its threshold concentration. That should be maintained, or that should be that is the important thing, that if the inefficient internalization occurs, the drug cannot be able to release properly, and the whole complex cannot be able to function properly. And in the fourth step,